Hello and welcome to my YouTube channel by Remote Class. Today's topic, as you can see, double fertilization. Double fertilization was discovered by Sergei Navashin in 1898 when he was experimenting on Lilium matagon and Fertilaria tenila. Now let us see what happens in double fertilization. Suppose this is the stigma of the pistil and pollen grains are present on the stigma. Now each pollen grain has two cells. One is a bigger cell which is known as vegetative cell or tube cell. And within the cytoplasm of the vegetative cell, there is a smaller cell which is floating within the cytoplasm of the bigger cell. This is known as the generative cell. Both the vegetative cell and the generative cell have nuclei. And the next step is pollen pistil interaction. Now, what is this pollen pistil interaction? The pollens will release some chemicals. At the same time, the stigma will also release some chemicals. Why do they release chemicals and what is the function of these chemicals? They check whether they belong to the same species or not. That means they check compatibility, whether they belong to the same species or not. Now, if they belong to the same species, then what will happen? There is a compatibility between the two. So the compatibility is there. That means further actions will take place and fertilization will take place. And if there is no compatibility, then what will happen? No fertilization will take place. That means these chemicals check whether they belong to the same species or not. If they belong to the same species, then they are compatible to each other. That means the further actions will take place, and as a result, the fertilization will take place ultimately. And if they do not belong to the same species, for example, there is a mango tree and on which the pollens of acacia have fallen, then what will happen? Acacia and mango are of different species, so there is no compatibility, there is no matching between these two. As a result, no further interaction will take place between these two and there will be no fertilization. Now let us see what happens during double fertilization. As you can see that this is the bigger cell or the vegetative cell and there is the generative cell present within the vegetative cell. The cytoplasm of the vegetative cell will start moving downward and it will form a tube. That means it will look like this. And as the cytoplasm will move downward, what will happen? The generative cell will also start moving downward. So this is the generative cell or generative nucleus, and this is tube nucleus. Now let us see the ovule structure. Ovule is present here. Ovule is surrounded by integument, and within the ovule there is nucellus. What is the function of the nucellus? It provides nourishment to the embryo. So this is nucellus. Now within the nucellus, the embryo sac is present. There are two cells here. These two cells are known as the synergids. And there is a cell present between the two synergids. It is known as the egg cell or egg nucleus, whatever it may be. And there are two small cells present at the center. These are known as the polar nuclei. And at the opposite end of the synergids, there are three cells present. These three cells are known as the antipodal cells. And this aperture is known as micropyle. One thing you must keep in mind that the synergids are always present at the micropyle end. Now, as the time goes, the tube starts moving downward further. And as it moves downward further, then what happens? These two cells also start moving downward further. And after some times, what happens? This generative cell starts dividing mitotically. That means the generative cell or generative nucleus goes on mitotic cell division. As a result, what happens? Two cells are produced. What are these two cells? So these two cells are produced. These two cells are known as the male gametes. That means two male gametes. So it will move further downward. From here, there is also another pollen tube is coming down. Now the question is why the pollen tube will not move towards this side or towards this side, but why does it move only through the micropiles? The reason is this: the synergids have filiform apparatus, some finger-like projections. These are the filiform apparatus. And they attract the pollen tube by releasing some chemicals. As a result, what happens? The tube moves towards the synergids. And during this time, this tube nucleus degenerated. That means it gets destroyed. So what happens? Only the two male gametes start moving downward. And ultimately what happens? It breaks the synergids and move further. Now look at this picture. You can see that there are two synergids which are marked with green color, which are present at the top, and in between the two synergids there is the egg cell, and at the middle there we have small tiny polar nuclei. As they are present at the center, so they are also known as the central cells. And at the bottom, that means at the opposite end of the synergids, three small cells are present. These are known as the antipodal cells. And as you can see that one of the pollen tubes is entering inside the synergids. It has broken down the synergid, and two male gametes are also entering inside. Now what will happen to these male gametes? One of the male gametes will fuse with this egg cell. So one of the male gametes will fuse with egg cell. This male gamete has n number of chromosome, that is haploid. Egg cell is also n numbered. So both of them togetherly form 2n zygote, which later will form the embryo. Now let us see what will happen to the second one. Now this second male gamete will fuse with these two polar nuclei. So the second male gamete, which is also n numbered, will fuse with two polar nuclei. That means each one has n number. That means n plus n plus n. There will be triploid or triple fusion. This will result in the formation of primary endosperm nucleus, or in short it is called PEN. So we are finding that 2 times the fertilization is taking place. So as the fertilization is taking place twice, the first male gamete, which has n number of chromosome, fuses with egg cell, which has n number of chromosome. As a result, zygote is formed, and zygote is 2n numbered. That means this is diploid. This process is called syngamy or fertilization, and this results in embryo formation later. Now let us see what happens to the other male gamete. The other male gamete, the second male gamete, which is also n numbered, fuses with two polar nuclei. Each of the polar nucleus has n number. That means n plus n plus n, or triploid nucleus is formed. This is called primary endosperm nucleus, or in short we call it PEN. So syngamy plus triple fusion makes the double fertilization. So this is all about double fertilization. If you like my video, then do like, comment and share, and don't forget to subscribe to my channel. Thanks for watching.